Welcome to the deep dive. Today, our mission is to explore the hidden, the microscopic machinery that allows plants to breathe, to grow, and to react to their environment.
Yeah, it is a truly wild topic.
It really is. We are pulling all of this from a genuinely fascinating 2026 paper published in the journal Science by Taxay and colleagues. It uncovers a biological switch that has been operating quietly on our planet for, well, hundreds of millions of years.
Right, way before we were ever here.
Exactly. But to really grasp the scale of what we're talking about today, I want you listening to imagine something for a second.
This is a good analogy.
Imagine that every single morning, thousands of tiny microscopic mouths on your skin just automatically opened up to, like, breathe in the sunlight. You don't have to think about it. You don't consciously flex a muscle or control it. The moment the sun hits you, these microscopic pores just fling wide open.
Which sounds like a horror movie, but for plants, it's just Tuesday.
Right. Plants do exactly this every single day, using specialized structures on their leaves called stomata. But the big question is, how on earth do they know it's morning? I mean, how does a leaf, which doesn't have a brain, right, it has no nervous system, no eyes, how does it sense the sun and mechanically open a tiny door?
It is a remarkable piece of research. And to really appreciate what this paper discovered, we need to first establish a fundamental difference in how life operates at the very foundational cellular level.
Okay, let's hear it.
When we think about animal cells, like your own muscle cells or neurons, we generally rely on sodium and potassium ions to regulate the electrical balance across our cell membranes.
So that is basically how our nerves fire, right?
Exactly. That's the animal way. But plants, they took a completely different evolutionary path early on. Plants use what we call a proton pump.
A proton pump. Okay, let's unpack this for the listener. What exactly is a plant proton pump doing on a physical level?
Specifically, we're talking about a massive protein called the plasma membrane H plus ATPase.
That is a mouthful.
It is, yeah.
Yeah.
But just think of it as a microscopic biological engine sitting right on the surface boundary of the plant cell. Its entire job is to consume energy. It uses ATP, which is basically the little battery packs of the cell, to physically and actively push protons out of the cell.
Wait, protons, so those are positively charged hydrogen ions, right?
Precisely. Now, because this pump is constantly shoving positive charges outside the cell, the inside of the cell becomes relatively negatively charged compared to the outside environment.
Oh, okay.
In biology, we call this state hyperpolarization.
So if I'm picturing this right, by shoving all those positive charges out into the void, the plant is essentially creating a voltage drop across its skin. It's literally charging a battery.
Exactly right. The plant cell charges itself like a literal battery, creating a really powerful electrical and chemical gradient. And, it uses that stored battery power to drive almost everything else it does.
Like what kind of things?
Well, it uses that gradient to pull in vital nutrients from the soil against their natural flow, to transport sugars throughout its stems, and crucially for our discussion today, it uses it to generate the immense physical water pressure needed to open those tiny stomata mouths.
Wow.
Yeah. So if you want to understand how a plant moves, how it expands, or how it breathes, you have to understand how this main proton battery gets turned on in the first place.
That is so cool. Okay, so I want to pause here for a quick recap for you listening, because we want to make sure we lock in these core concepts.
Good idea.
So bullet point 1, plants don't use sodium like animals do. They use a proton pump to charge their cells like a battery. Bullet point 2. This process is called hyperpolarization, which is just a fancy word for shoving positive charges outside to make the inside negative. And bullet point 3, this battery power is what physically forces the plant's tiny mouths, the stomata, to open.
Perfect summary.
So here's a question for you listening to ponder as we move to the next part. Next time you look at a houseplant, can you picture that invisible electrical hum happening on its surface? Because it is constantly running that battery. So that naturally begs the question for our next subtopic. If the pump is the battery, where is the ignition switch? I mean, how does the plant actually turn it on?
Well, for decades, scientists have studied this specific proton pump. In the standard model plant used in labs, which is called Arabidopsis, the pump is known as AHA1. And scientists knew that the pump is regulated by a very common biological mechanism called phosphorylation.
Phosphorylation. Okay, if you are listening and don't spend your days in a biochemistry lab, Just picture this as a molecular sticky note.
That's a great way to think of it.
Right. It's the process of taking a phosphate group and just slapping it onto a protein. But it's not just a visual tag, because phosphate groups are highly negatively charged. So when you attach one to a protein, that negative charge repels other parts of the protein, which physically forces the whole machine to fold into a new 3D shape, and that activates it.
Exactly right. It physically changes the machinery. Now, the HT1 pump has a long molecular tail at its end. Normally, this tail just folds back onto the main body of the pump and acts like a brake.
So it's keeping itself turned off.
Right, but we call it an auto-inhibitory domain. To release that brake and let the engine run, the plant needs to attach two specific phosphate sticky notes to two very specific spots on that tail.
Here's where it gets really interesting. Think of releasing this brake like launching a missile from a submarine.
Oh, I love the submarine analogy.
Yeah, because you can't just press one button, right?
Yeah.
You need two different operators standing across the room to turn two separate keys at the exact same time.
That is exactly what happens. The first keyhole is an amino acid on the tail called threonine 948. We've actually known about that one for a while. We knew it gets phosphorylated when the plant senses blue light.
Okay, so that's the first key.
Right. But the second keyhole, threonine 881, has been a massive mystery in the botanical world. The pump will absolutely not turn on its highest gear unless threonine 881 is also phosphorylated. But literally nobody knew who was turning that second key.
Until this paper.
Exactly.
The researchers finally caught the mystery operator red-handed.
Yes, they did. And what they discovered is that it's not just a single operator tuning the threonine 881 key. It's actually a coordinated team effort. Through some really complex genetic screening, they identified a specific class of proteins called Raf-like kinases.
Wait, let's define kinase real quick. A kinase is simply a type of enzyme, right? Like a biological machine whose entire job is to slap those phosphate sticky notes onto other things.
Spot on. But the plant doesn't use just one kinase for this job. It uses what we call a heterocomplex.
Heterocomplex.
Yeah, which is basically a paired team consisting of a C5-type Raf kinase and a C7-type Raf kinase.
Okay, I'm picturing this like a molecular relay race. You have the first runner, the C5 kinase. It starts the sequence. The C5 runs up and passes the baton, meaning it transfers the activating energy, to the next runner, which is the C7 kinase.
That's right.
And once the C7 kinase has the baton, it turns around, rushes to the proton pump, and directly flips the Thrad81 switch. C5 passes to C7, and C7 turns the key.
That is exactly the sequence.
But that makes me wonder, why? Why have a molecular relay race with two distinct kinases instead of just having one protein do the job directly? It seems like an extra unnecessary step.
Well, this raises an important question about biological efficiency. And the answer is all about modular customized control. Because if you have just one kinase doing the job directly, you have a very simple, rigid system. It's always on or always off in the exact same way. But by having a two-part tag team, the plant can mix and match. The paper demonstrates beautifully that different tissues in the plant use different C5 and C7 pairs.
Oh, I see. So it's like having a universal engine, the C7 in the pump, but being able to plug in different specialized sensors, the C5s.
Right.
So it depends on whether you are operating in a dark root underground or a rigid stem or a delicate leaf out in the sun.
Exactly. It gives the plant incredibly fine-tuned, tissue-specific control over its main battery without having to reinvent the core machinery for every single cell type.
Okay, let's pause here for another quick 5-minute recap for the listener to lock in this relay race concept.
It is good.
Bullet point 1, the proton pump is kept off by a brake on its tail. Bullet point 2, you need two keys turned to release the brake, threonine 948 and threonine 881, and bullet point 3. The second key is turned by a two-part relay race made of a C5 kinase passing the baton to a C7 kinase.
Noted.
So my question for you listening is this. Think about the machines in your own life.
Yeah.
Wouldn't it be easier if everything used a universal battery but had custom sensors? Nature figured that out millions of years ago. So, moving on to the next part, we have this elegant two-part relay race. But I want to know what happens when things go wrong.
The mutant studies.
Yes, because if you have this intricate sequence just to turn on the plant's main battery, what happens if one of the runners drops the baton?
To answer that, the researchers looked at the main body of the plant, the flat leaves. In the leaves, the plant uses a specific C5 kinase called Raf36.
Okay, so Raf36 is the starting runner in the leaf tissue.
Right. And to prove that this relay race actually matters to a living, breathing plant, the researchers used gene editing to create mutant plants. They completely deleted the gene that creates the Raf36 kinase. These mutants are referred to in the paper as Raf36-1 and Raf36-2.
So they basically took a genetic scalpel and just completely removed the first runner.
Exactly.
What did these mutant plants look like?
The visual symptoms were dramatic. When you looked at these plants growing in their pots side by side with normal, healthy plants, the mutant suffered a 40% reduction in overall leaf area.
40%. That is massive.
Yeah, their leaves were tiny and shriveled. And when the scientists put those tiny leaves under an electron microscope, they saw that the individual cells making up the leaf's surface, what we call the pavement cells, were physically smaller.
Why were they smaller?
Because the main proton battery couldn't be turned on fully. The cells couldn't pull in enough water and nutrients to physically swell and expand. The plant simply couldn't grow.
That makes intuitive sense. But let me play devil's advocate for a second.
Okay, go for it.
We have a stunted plant missing a protein called Raf36. How do we know for absolutely sure that the plant is tiny, specifically because that Thray81 battery switch wasn't flipped? I mean, maybe the plant is just generally sick and dying because you ripped a major protein out of its DNA.
That is the exact critical thinking that scientists applied. Because, correlation is not causation.
Right.
First, they used an incredibly precise technique called phosphoproteomic analysis to see what was happening at the chemical level.
Okay, for the listener, phosphoproteomic analysis just means they effectively ground up the plant tissue, put it through a mass spectrometer, and scanned all the proteins to see which ones had those phosphate sticky notes attached.
Exactly. And in the stunted mutant plants, the first keyhole in the pump, 3948, was perfectly fine. It was phosphorylated normally.
Okay.
But the second keyhole, 3881, the phosphorylation signal dropped by 90%.
Wow, so they proved the baton was definitely dropped. The second key wasn't turned. But how did they prove that unturned key was the actual cause of the stunted leaves?
To prove causality, you have to fix the specific broken part and see if the engine runs again. They used an elegant genetic trick. They took those stunted mutant plants that were completely missing Raf36, and they edited their DNA again to alter the 3881 keyhole itself.
Okay, how did they alter it?
Well, threonine, the amino acid normally sitting at spot 881, is electrically neutral. It needs the negative phosphate added to it to work. But the scientists swapped out that threonine for a different amino acid called aspartic acid.
Wait, aspartic acid. Doesn't that naturally carry a negative charge on its own?
It does. By swapping in aspartic acid, which is called the T881D mutation, they permanently placed a negative charge right where the phosphate sticky note was supposed to go.
Oh, that is brilliant. You are essentially hot wiring the ignition.
Yes.
You're fooling the proton pump into thinking the kinase has already flipped the switch forever without the kinase even being there.
Exactly. They completely bypassed the missing Raf36 runner, and the result of that hot wiring, those previously stunted plants grew back to 100% normal size. Their leaves expanded, perfectly full and healthy.
That is so satisfying.
Right. And to double check, they also swapped the switch for an amino acid called alanine, which is neutral and cannot be phosphorylated, creating a permanently off switch.
Let me guess, those plants stayed small.
Those plants stayed permanently stunted. This proved definitively that the entire reason Raf36 exists in the leaf is to flip that single Thray81 switch and drive cell expansion.
Okay, time for another quick recap.
Let's do it.
Bullet point 1. Removing the Raf36 runner makes the plant 40% smaller because the cells can't swell up. Bullet point 2. A mass spectrometer proved the Thray881 key wasn't getting turned. Bullet point 3. They hot-wired the pump by swapping in aspartic acid, which has a natural negative charge, and the plant grew back to normal.
Great recap.
And my question for the listener is, if you were a scientist, would you have thought of that aspartic acid hot-wiring trick? It's basically hacking the plant's hardware. So we've solved how the flat pavement cells of the leaf expand to grow the main body. But what about those tiny mouths we talked about at the very beginning? The stomata.
Right, the breathing pores.
Yeah, how does this exact same proton battery mechanism make those specific pores open when the sun comes up?
That requires us to look closely at the guard cells. The stomatopore itself is actually just an empty hole. It is surrounded by two specialized balloon-like cells called guard cells.
Sure.
Remember how we said the plant uses different C5 sensors in different tissues?
Well, it's the modular system.
Yeah. Well, in these guard cells, the plant does not use Raf36. It uses a totally different C5-Raf called HT1.
Okay, so HT1 is the starting runner in the guard cells. Who does HT1 pass the baton to?
It passes it to C7-Rafs, known as CBC1 and CBC2. And here is where we truly see the computational software of the plant at work.
Ooh, computational software.
Yeah, because this isn't just a physical relay race anymore. It is functioning as a biological logic gate.
Like in computer programming.
Exactly. A plant cannot just leave its stomata open all the time, or it would lose all its water to evaporation and die. It needs to only open them when conditions are perfectly right for photosynthesis.
Right. It's running a biological if-then algorithm inside the leaf. To understand how that code runs, we should look at a leaf's daily routine. Let's start at midnight. It's pitch black outside. What is happening chemically inside the leaf?
At night, there's obviously no sunlight, so the plant cannot perform photosynthesis. However, the plant cells are still alive, meaning they are doing cellular respiration just like our cells do, which produces carbon dioxide as a byproduct. Because the stomata doors are closed to save water, all that CO2 is trapped. It builds up, making the internal CO2 concentration of the leaf very high.
And from what I understand from the paper, this high level of trapped CO2 triggers an inhibitor protein.
Yes. High CO2 inside the leaf causes a heavy-duty inhibitor complex called MPK412 to physically clamp down onto our C5 kinase, HT1.
Oh, so it just grabs it.
Right. MPK4 grabs HT1 and prevents it from passing the baton. It's a complete system lock. The relay race is halted at the starting line. No signal passes to CDC1, the thread 81 switch stays off, the proton battery stays off, the guard cells stay deflated, and the stomatophore remains sealed shut.
I love this setup. Let's use an analogy here for you listening. Think of the plant cell like a factory at night. MPK4 is the night watchman.
That's perfect.
He is physically holding the front doors of the factory shut. He is holding back the morning opening crew, which is HT1, and the watchman has strict orders like he will not let the opening crew do their jobs as long as the factory floor is cluttered with leftover, unused inventory.
Which in this case is that massive buildup of CO2.
Exactly. So the sun finally peaks over the horizon. What happens?
What's fascinating here is the precise mechanical sequence of events that follows. Red light from the morning sun hits the surface of the leaf. This instantly kicks off the machinery of photosynthesis inside the cells. Photosynthesis acts like an aggressive vacuum cleaner, rapidly sucking up and consuming all that internally trapped CO2 to manufacture sugars.
The factory machines are turning on and rapidly clearing out the old inventory.
Exactly. And as that CO2 concentration plummets, the environment changes. The MPK4 inhibitor protein physically changes its shape in response to the low CO2, and it detaches from HT1.
The watchman clocks out.
The watchman clocks out. Now, HT1 is finally free. It instantly activates its partner, CBC1. CBC1 rushes to the cell membrane and flips the thread 81 switch on the proton pump.
And the battery turns on.
The main battery roars to life. It starts pumping out protons, hyperpolarization occurs, and positively charged potassium ions are drawn rapidly into the guard cells.
And wherever salt or ions go, water follows by osmosis.
Exactly. Basic chemistry.
The inside of the guard cells becomes so packed with potassium that water from the surrounding tissue just rushes in to dilute it. The two guard cells swell up with extreme water pressure, bending outward like two overinflated long balloons, and the stomatophore in the middle bursts open for business.
Really is beautiful.
Let's walk through that biological logic gate one more time because it is incredible. If red light hits the leaf, meaning photosynthesis starts, and if low CO2 is achieved, meaning the internal inventory is cleared, then the watchman MPK4 detaches, HT1 activates, 3881 flips, and the stomata open. It's a literal functioning Andy gate made entirely of interacting proteins.
It is stunningly elegant. And the researchers didn't just guess this sequence. They proved it visually using a highly advanced imaging technique called FLIMFRET.
Wait, FLIMFRET, what is that?
In simple terms, FLIMFRET utilizes fluorescence lifetime and energy transfer. If you tag two different proteins with glowing markers, and those proteins get incredibly close to each other, meaning they are physically touching, one protein will actually steal the glowing energy from the other.
Oh.
Yeah, by measuring that energy theft under a microscope in living cells, the scientists could physically watch the MPK4 watchman clamping onto HT1 and then watch them separate when the CO2 dropped.
That is insane that we can watch that happen in real time.
And just like with the leaves, when they used the T81D hot wiring trick on the guard cells, the stomata stayed wide open regardless of the CO2 levels, bypassing the broken sensors entirely.
It is just a masterclass in proving a biological pathway. Let's do another recap before we move to the evolutionary history.
So good.
Bullet point 1, the stomata use a logic gate, Bullet point 2, high CO2 at night makes MPK4 act as a watchman, keeping the doors locked. Bullet point 3, morning sun triggers photosynthesis, clears the CO2, the watchman leaves, and water rushes into the cells via osmosis to pop the doors open. And my question for you listening is, have you ever thought of a plant as a computer running code? Because that's exactly what an if then logic gate is. So we've seen this incredible protein software running flawlessly in Arabidopsis, which is a modern, complex flowering plant. But as I was reading this paper, a huge question popped into my mind.
Which is?
When exactly did nature write this code? Did plants just invent this highly specific relay race recently to manage their stomata?
If we connect this to the bigger evolutionary picture, the answer is no. And this is where the paper takes a brilliant historical turn. The researchers didn't just look at modern complex plants, they went all the way back to the very base of the terrestrial plant family tree. They sequenced the DNA of a liverwort, specifically a plant called Marchantia polymorpha.
Okay, if you aren't familiar with liverworts, they are some of the most ancient land plants on Earth. They don't have true stems or deep roots. They look a bit like flat green ribbons crawling on wet river rocks. They have been surviving on this planet for hundreds of millions of years, long before the first dinosaur took a breath, long before flowers or seeds even existed.
Right. They're an evolutionary time capsule. And when the researchers looked deep into the genome of this ancient liverwort, they found the exact same modular system. They found a single C5-Raf gene and a C7-Raf gene.
Wow.
They used modern CRISPR gene editing to mutate the liverwort's C5 gene, breaking the relay race. And the exact same thing happened that we saw in the modern plant. The liverwort's main body became severely stunted and small.
Incredible.
When they ran the chemical tests, they found that these ancient liverwort kinases were targeting the exact same light-induced switch on the liverwort's primitive version of the proton pump.
But wait, I have to push back here because there's a fascinating biological contradiction in the source text that caught my eye.
Oh, I know what you're going to say.
The paper specifically notes that while the distant ancestors of terrestrial plants likely acquired stomata, liverworts actually lost their stomata during evolution. They don't have them. So if liverworts don't even have these tiny mouths to open, why are they carrying around this exact same complex proton pump switch and kinase relay race? What's it doing?
That observation is perhaps the most profound realization in the entire paper. It proves that the C5-C7 relay race did not evolve for stomata. It evolved hundreds of millions of years ago as the fundamental core operating system for all plant life.
Just to turn on the main battery.
Exactly. Turning on that main proton battery is absolutely essential for basic cell expansion, pulling nutrients from bare rock, and sheer survival. Eons later, as higher plants evolved complex leaves and those new tiny mouths called stomata, they didn't bother inventing a brand new battery system.
Evolution is the ultimate recycler.
It really is. They just took that ancient pre-existing pump switch and hooked it up to a brand new sensor, the MPK4 and HT1 light-induced CO2 sensor, to perfectly control their new mouths.
Unbelievable. It basically just repurposed the base code for a new app. So before we wrap up, our final mini recap.
Right?
Bullet point 1. Liverworts are ancient plants that don't even have stomata. Bullet point 2. They still use the exact same light-induced switch to grow. Bullet point 3. Modern plants just recycle this ancient battery switch to run their modern breathing force.
Excellent.
And my question for the listener, does it blow your mind that the code running the oak tree in your yard is the exact same code that was running on wet rocks before the dinosaurs?
It really puts time into perspective.
It does. So what does this all mean for us today? We started this deep dive looking at a microscopic break on a proton pump. We unraveled a relay race with kinases. We watched how a leaf grows. And we marveled at how guard cells act as a biological logic gate to open to the morning sun. We've traced a software program running across the entire history of the plant kingdom. But beyond this being a staggering fact of nature, what is the real-world application? Why should you, the listener, care about exactly how light-induced gets phosphorylated?
Because it is incredibly relevant to the future of our survival.
That's a big claim.
It is. But this isn't just academic trivia. We are facing a rapidly accelerating global climate crisis and a booming human population that desperately needs to be fed. The paper explicitly notes that manipulating these specific plant proton pumps has already been shown in early trials to successfully enhance crop productivity.
Right, because if you control the master battery, you control how the plant interacts with the world.
Exactly. By understanding exactly how to hotwire this 381 switch, like we saw with the aspartic acid trick, or by genetically tweaking the specific C5 sensors that trigger it, we could engineer agricultural crops that do incredible things. We could program crops to grow larger leaves, capturing more sunlight and creating massive increases in food yield.
Wow.
More importantly, we could program their stomata to snap shut much faster at the very first molecular sign of a drought. Right now, crops lose massive amounts of water before their stomata fully close. If we tune the sensor, we save the water, helping crops survive extreme weather.
Which is going to be vital.
And we could even engineer stomata that open wider and stay open longer under safe conditions to aggressively capture more carbon dioxide from the atmosphere. By decoding this ancient biological machinery, we're literally gaining the instruction manual to potentially help feed the world and stabilize our climate.
That is massive. We are moving from merely observing nature with microscopes to fluently speaking its molecular language. And that leaves us with a final provocative thought for you to ponder long after this deep dive ends. We just spent all this time exploring how a plant uses interacting proteins to build a literal, functioning computer logic gate. If sunlight and a low CO2, then open stomata. A completely blind, brainless leaf outside your house is doing complex data processing every single morning. It makes you wonder what other intricate biological software programs are running silently in the greenery right outside your window right now. And if we are finally learning to decode their protein logic, how long until we can write our own code? How long until we can program a forest the exact same way we program computers? Think about that the next time you watch a leaf twisting to catch the morning sun. Thanks for taking this deep dive with us.